Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 4, let's be talk about separation and isolation of DNA fragments. Separation and isolation of DNA fragments. The cut of the DNA, the restriction endonuclease result in the fragments of DNA. So when cut the DNAs, after it is to get forms as what? Small DNA fragments we are formed. Let's here these fragments separated by technique is called as gel electrophoresis. So it is in gel electrophoresis. So after the cut the total DNA fragments they having small some large is present and some small is present like. So how do they get separated? Here the long DNA fragments, medium fragments and small fragments, how to get they are separated with help of only gel electrophoresis. Since DNA fragments are negatively charged molecule, they can separate it, the forcing of move towards an anode under electron, the electric field through as in a medium or matrix. So mostly already you know the DNA fragments as in what? Always negative charge, right? So then what happened? The mostly the total DNA fragments get transferred into as in a anode region. Transferred into anode region. Nowadays mostly commonly the matrix agarose which is a naturally polymer extract from as in seaweeds. So in this total in this the mostly in this gel electrophoresis machine, in this complete the gel is present. That gel we are extract from where? We are extract from as in their seaweeds. Seaweeds. The DNA fragments separated, they are resolved according to their size. They are completely the sieving the effect the provide by as an agarose gel. So with the agarose gel method, here when wells we are completely allow it as in DNA, what happened? The DNA fragments to get separated. Okay na? Next, hence the smaller fragments, once the smaller the fragment size, the further it moves. Here see in that diagram, the mostly the smaller fragments, medium fragments, like they get separated as in a easily. Okay na? So it is in a part of what? In a completely the gel electrophoresis method, how do the DNA fragments they are separated. After the separation, after this total DNA fragments, since smaller came and medium bands came and large bands also they get came. Then after the DNA fragments can be there visualized after the staining the DNA with their compound known as an ethidium bromide which followed by as an UV radiation. Without a DNA can't be, the pure DNA fragments can be visualized light without any staining. So first after we get as a DNA species without stain, it is not possible. So after in this total DNA fragments, you add with as an ethidium bromide solution. Ethidium bromide solution. After then to get exposed to as an UV light or UV radiation, then only the total the DNA fragments to get appear as an orange bands. Okay, so you can just see the bright orange color bands of the DNA, the ethidium bromide, the stained gel, they are exposed to UV light. So when you are add with as ethidium bromide, then appear as in what? You can keep UV light, under UV light, they get completely appear as DNA fragments or orange color bands we are noticing. The separation bands of DNA cut from as an agarose gel and extract from as an gel pieces. So after to get extract from as an gel pieces, this step known as an elution process. This step is called as what? Elution process. Means after the DNA get separated, that process is called as what? Elution process. Okay. Let us. After the DNA fragments, they purified with a way they used construction recombinant DNA which joined with cloning vectors. After to develop this total DNA, we clone with as well into a vector. Okay, now this is cloning vectors. Now the plasmids and bacteriophages have the ability to replicate within the bacterial cell independent of control of chromosomal DNA. So mostly the plasmids has an ability to get produced as a multiple copy of the chromosomal DNAs. For example, here has only one DNA fragment is present, right? So here the multiple copies of the DNA fragments, how to get forms? See here with help of the any one vector only. Okay, And the bacteriophages, because they are high number of per cell, high vary the copy numbers, they are a genome within the bacterial cell. So mostly the bacterial genome cell to get produces in multiple copies. Some plasmids may have only 1 or 2 copies per cell, whereas other may have in a 15 to 100 copies per cell. So one they having in 1 to 2, or some they having as in 15 to 100 copies of the cells get produced. Okay, next, their number can go even higher. So if in case they get go to as in the number of copies, they get go to as a number of copies. If they able to link alien DNA within as in the bacteriophage or plasmid, we can there multiply number of equal the copy number or plasmid bacteriophages. So if in case one we require, it is get produced in one. You want as in multiple copies, it is get easily to get produced in multiple copies. Like vectors used at present at engineered such as way, they are help to as easy link to foreign DNA to selection of recombinant from non-recombinants. So recombinant DNA molecules you can easily to get developed as any the process of in this vectors only. Okay, next, after some following features required for the facilitated cloning into as a vector. Here we are completely we require as any some facilitative cloning vectors. Okay, let's see. First one is what origin of replication means, simply called as ORI, origin of replication. Okay, let's origin of replication ORI. This is the sequence from where the replication start, and any piece of DNA linked with as any the sequence can be there made to the replicates within a host cell. So for example, here has any in that host cell is present. So we are get to get any the vector DNA get inject here, what happen? It is to get produces as any their multiple copies. Get produces what? In a their multiple copies of the DNAs are produced. And the sequence also the responsible for the controlling the copy number of linked DNA. So here the most the sequence also it is responsible for which purpose? It is a controlling of the copy of number linked DNA. So how many the copy of numbers be required? Okay, so if one wants to recover many copies to target DNA, should be there cloned into as in a vector. Those organs support a high copy number. So here, when the number of DNA fragments we require, they're easy to get origin with help of as any what this ORI site. Okay, that is next selectable markers. Second one is what? Selectable markers. In this addition of ORI, the vector is required selectable markers, which has a help to identifying and eliminating non-transplants and the selectively permitting as in a growth of the any transplants. So which one is required? That is they get completely they get the recombinants and non-recombinants means identifying and eliminating with help of only for selectable markers. Next, if the transformation is in a procedure through the which the piece of the DNA introduced into as a host bacterium. So here which the DNA is required, that is to get introduced into as a host organism, then what happened? They get producing as in a their multiple copies in the process of in a their subsequently section process. Next, normally the genes encoding the resistant to antibiotics such as in ampicillin, chloramphenicol and tetracycline and kanamycin. So here there are some antibiotic resistant genes as present, like as in ampicillin, tetracycline, kanamycin, chloramphenicol. Like these are completely called as what? In a antibiotic resistant genes. Okay, etc. Like are they considered as any useful selectable markers? Escherichia coli. So the Escherichia coli they're using as in this kind of the antibiotic resistant genes. The normal E. coli cells do not carry as resistant against any of these antibiotics. So mostly that Escherichia coli does not carry any resistant against the antibiotics because they are producing as any their antibiotics here only. Here next third one is what? Cloning site. In order to link the alien DNA into vector, it needs to have very few in probably the single recognition site. So only one single recognition site we get present. For that, in commonly used as a restriction enzymes. So here the restriction enzymes to get producing as a number of their sites. Presence of the more than one recognition site within their vector will be generated as in a several fragments. So most recognition sites they are present many. Only at the site what happened will be generated as in a several fragments. Next, which will be their complicate the gene cloning process. So mostly in the gene cloning process, here they get completed, the number of cells will get produced. Next, the ligation of alien DNA is carried out a restriction site in the presence of one or two antibiotic resistance. So mostly when you observe here, it is get produced as in one or two, the restriction sites will also they are produced. Here, for genes, their example they given here to ligate it. Here, for example, you can ligate it foreign DNA at the BAMH1, the site for the tetracycline, the resistance here, the genes for example here ligated for foreign DNA, the BM, BAMH1 means H1 site, it is a tetracycline resistant gene, it is a vector PBR322. So once you observe to as any a PBR322, in this site they having as what tetracycline is present. Here, the recombinant, the plasmid will be lost the tetracycline resistant due to as an insertion of foreign DNA into can be the still they selected as an for going as a non-recombinant ones by as any the plating that in a transformants. So mostly here they having as what only tetracycline is present. Tetracycline it is not growing as in that amphicyclian region. Okay, now see in this process here it is only one the plating of the transformants on the tetracycline containing medium. So only they're growing as what in only for tetracycline medium. The transformant growing on amphicyclian containing medium or they are transformed they're transferred on the medium containing the tetracycline. So here amphicyclian is present, but amphicyclian that is not growing as in that medium. But the recombinants will growing in where only for in a amphicyclian containing medium, but not as in containing tetracycline. So mostly the recombinants growing as where the recombinants growing as in a only for in a amphicyclian medium, not a tetracycline. Going as in only for in a amphicyclian medium only. Next, but here, but the non-recombinants will grow as in a medium containing the both their antibiotics. So here is non-recombinants they growing as in a both antibiotics. In this case of the antibiotic resistant genes help the selecting the transformants, whereas in another antibiotic resistance genes they completely inactivated due to as an insertion of LNDNA into as a help of in a selection recombinants. So mostly they are completely where the antibiotic resistant genes is present, they go to as what? They go to as in a their inactive condition for insertion of alien DNA with the help of what? Recombinants. Okay, next. Now the selection of recombinants due to as an inactivation of antibiotics is in cumbersome. The produce as it becomes requires as in their stimulant the completely simultaneous plating as in a two plates having different antibiotics. So when we are completely growing as in the different plates, they growing as what in a different antibiotics or to get growing. Therefore, their alternate selectable markers have been developed which has in differentiated recombinants from non-recombinants on the basis of the ability to produce the color and the presence of in the chromogenic substances. So which one they are completely here recombinants or non-recombinants they completely they are how to get separated with help of the formation of as in a their color substance. If in case color substance is produced or not producing, okay now. So that is how do the colors will be get forms because they are they having as what chromogenic substance is present. Okay, next after in this recombinant DNA, in this recombinant DNA is incision with as in a coding sequence of enzyme like that is beta galactose. So here the when their enzyme they are introducing that is beta galactose. So once their beta galactose will be there introduced, what happened? The result their inactivation of the gene synthesizing of the enzyme. So here completely the synthesizing will be there inactivated, which is refers to as an institutional inactivation. So when they are synthesizing it will be stopped, that is called as what? Institutional inactivation process. In this process, chromogenic substance gives as in blue color colonies with if the in a plasmids bacteria does not have any inside. So here when they are complete, this process will be going on what the color colonies we are noticing. Okay, what type of color they give as in a completely blue color. So because they having as what chromogenic substance is present. Okay, presence of the initiation of result into as an institutional inactivation of the in a beta galactose gene, and the colonies does not produce any color. This is identification of as any recombinant colonies. So where the colonies do not have produce any color, that we are identified is what, that is completely called as a recombinant colonies. That is in a recombinant colonies. Remaining we discuss in next class. Thank you for watching. Please share and subscribe. Thank you.